Okay, so we're going to be doing a little bit of DNA. Okay, a lot of DNA, not a little bit of DNA, but this will be preparing you towards the topic of genetics. Before you're able to do your monohybrid crosses and your dihybrid crosses, you should be able to go through the entire chapter of DNA with probably your eyes closed. So we're going to go into DNA, but before going into DNA, we need to first recap the cell. So specifically, we're going to work on the animal cell, and on the animal cell, the parts that you need to know are the cell membrane, the mitochondrion, the ribosome, the cytoplasm, and your nucleus, and probably your nucleolus as well, but not exactly that important. And you also need your nuclear membrane. So let's do a bit of a recap. We remember the cell membrane. We remember that it is semi-permeable, meaning it selects which substances can go into and out of the cell. This we remember. Then the mitochondrion, we remember this is the powerhouse of the cell. For those who remember it quite well, it's the powerhouse. It's where the energy is generated and all of it, isn't it? So that's where the energy comes from, and it's also where respiration takes place. Okay, so which you all remember, respiration takes place in the mitochondria. All right, and then ribosome. Ribosome is an organelle that is located in the cytoplasm, and you remember the cytoplasm is this blue fluid right there, and the cytoplasm is what carries all these other organelles. So the cytoplasm is what makes sure that the cell does not dehydrate, so it's fluid like it has water and all that, so it keeps it hydrated. Right, so now this ribosome, we remember that this is the site where protein synthesis takes place. Sorry about that. This is the site where protein synthesis takes place. Protein synthesis, what was protein synthesis? Well, it's a process where we generating proteins through amino acids. But let me not bombard you with a lot of things. All you need to know is the basic parts of the cell, which I have highlighted out. Alright, now what is it in DNA that we need to know? Well, firstly, we need to remember the structure of the chromosome. The chromosome is built up of three things: two chromatids, so 1 and 2 chromatids, and then in the middle it has something that binds the two chromatids together, and this is called the centromere. Right, so 2 chromatids, and all these are binded by a centromere. All right, and then this chromosome, it has segments on it like this. Right, and each of these segments, they code for a specific characteristic. Right, and now these segments that are kind of like leather like, so these segments are what you're going to call your genes. These are your genes, not the ones you wear, the genes. All right, so that's where you're going to find your genes. And remember, these are what code for, this is basically what codes for a specific characteristic. You need to remember that they code for a specific characteristic. Okay, and your centromere, it's what binds the two chromatids together. They love the question in the NSC question paper, where they'll give you an incomplete drawing of this with incomplete labels, with probably some letters, and they would ask you to label the unknown letters. So you should be able to identify that that's a chromatid, and in the middle you have a centromere, and the specific segment there is called a gene. All right, then we have what are called nucleic acids. Nucleic acids, you probably heard this from DNA, right? So we have two types of nucleic acids. We have DNA and we have RNA. Okay, for those who remember, DNA stands for deoxyribonucleic acid. Again, that is deoxyribonucleic acid. All right, and RNA stands for ribonucleic acid. Okay, so now these two are the two types of nucleic acids you have, and they're each different, and they are both used for different things in the human genome. So all nucleic acids are built up of what are called monomers. Okay, and these monomers are called nucleotides. What are monomers again? Monomers are building blocks. Okay, these are building blocks. If I want to build a house, I need some bricks. So the monomers of the house is the bricks. Hope that is easier to understand that way. So nucleic acids are built up of nucleotides. Okay, we always need to remember that. Then how does a nucleotide look like? Remember, this is how both DNA and RNA typically look like. We'll go over their differences soon. So now the nucleotide is L-shaped. Okay, basically that's the representation you have. So now that it is L-shaped, you have a component that goes there, and one there, and one there. Okay, so remember, if you put monomers together, then you're going to have a nucleic acid, right? So if you have multiple nucleotides, you're going to have a nucleic acid. So on the top here, you're going to have a phosphate group, okay, or a phosphate ion. So that's your phosphate, hence it is shown there with a P. And then here, you're going to have a sugar. Alright, you're going to have a sugar. And then right there we have what is called a nitrogenous base. You just need to know that it is L-shaped and it has these three components. So this is how a typical nucleotide looks like. We're not yet going into the specific on whether it is DNA or RNA, right? Okay. So now what is the difference between DNA and RNA? Now that we know how a nucleotide looks like, then we can go into the actual stuff. DNA contains deoxyribose sugar, while RNA contains ribose sugar. Back there. Here. So this was our nucleotide. So DNA, in this part here, it has deoxyribose sugar, but RNA has ribose sugar. Okay. So that is the difference when it comes to the sugar molecule. Right. Okay. Then it is said that DNA is double-stranded while RNA is single-stranded. What does this mean? Well, starting off, DNA is like a ladder. Alright, it is like a ladder. There. So you've got the backbone and you've got your nitrogenous bases here. You're probably like, what? But anyways, there at that point, as we know, at that point there, right there, that's where we have our phosphate group, and then there you've got your sugar, right? The sugar goes there, sorry about that. So you've got your sugar goes there, and in the middle there you're going to have a nitrogenous base. Right, you're going to have a nitrogenous base. So, and it goes on and on. So phosphate, sugar, nitrogenous base, phosphate, sugar, nitrogenous base, phosphate, sugar, nitrogenous base. And that is just one side of the strand. This is DNA, remember, there's just one side of the strand. And then on the other side, they are kind of symmetrical. On the other side, it's another phosphate, sugar, nitrogenous base, phosphate, sugar, nitrogenous base, and so on and so forth. Alright, so hence they say it's double-stranded. You've got a strand on one side and another strand on the other side. But RNA only has a single strand. It's an incomplete ladder. Okay, so it's just like that. Alright, so it's quite similar. Phosphate, sugar, nitrogenous base, phosphate, sugar, nitrogenous base, phosphate, sugar, nitrogenous base, and so on and so forth. Alright, both nucleic acids have nitrogenous bases. There are 4 nitrogenous bases, but we're going to go through them in detail. Alright, so now nitrogenous bases, it's this part. There are 4 types of nitrogenous bases that we have. Alright, we have adenine, we have guanine. Now I'm listing the nitrogenous bases found in DNA. Remember that these are nitrogenous bases found in DNA. So we've got adenine, guanine, cytosine, and last but not least, we have thymine. Now we remember that DNA is said to be double-stranded, so there are two parts of the ladder, as we remember. So now it's this part, this middle portion here, that we have your nitrogenous bases. We remember that you have a phosphate group, you've got a sugar molecule, and then right there you've got a nitrogenous base, and then also on the other side you've got a phosphate, a sugar, and a nitrogenous base. So now there's a bond between this, there's a bond that is in between these nitrogenous bases from the two separate strands, and the name of that bond is called weak hydrogen bonds, WHB. Okay, so those are your weak hydrogen bonds. So now the way in which you pair your nitrogenous bases has to make sense according to the complementary base template. Okay, so it has to make sense. You cannot just take adenine and pair it with any. There is a specific way that you have to pair them. Okay, and the best way to remember this is cute girls are all taken. CGAT. Okay, cute girls are all taken. Cytosine goes with guanine, adenine goes with thymine. Always remember that. Alright, so when you, for instance, have cytosine there, then your guanine will be there. Alright, I hope it makes sense that way. When you have adenine there, you have thymine there. They cannot be mixed up in any other different way. So cytosine goes with guanine, and guanine goes with cytosine. Okay, and adenine goes with thymine, while thymine can also go with adenine, and we call these complementary bases. Alright, so that was DNA. And then RNA also has 4 nitrogenous bases, but luckily they are kind of the same as the ones of DNA. So RNA, the nitrogenous bases of RNA are adenine, guanine, cytosine. Now instead of thymine, RNA will instead have what is called uracil. Alright, it will have uracil. Now when it comes to the complementary bases of RNA, it's no longer CGAT, but it's now CGAU. Pretty sick, isn't it? But that was quite simple. So that those are the nitrogenous bases, and that's how you pair them all together. Alright, so now going back deeper into DNA. Right, so DNA, the shape of DNA in nature is said to be double helix, and double helix means that it is winded up into a helix spiral like structure, like that. So it is double helix like that. Okay, always remember the shape is in nature is a DNA is double helix. Always remember that. Then looking over at DNA and RNA, firstly with DNA, there are 3 types of DNA. You will need to write those down, but those you'll find in the notes coming up. So the 3 types of DNA that you have are going to be nuclear DNA. Nuclear DNA is the DNA that we find in the nucleus. This we know, right? So it's nuclear DNA. Then you also have DNA found in the mitochondria. Okay, and this we call mitochondrial DNA. Alright, and then we have DNA found in the chloroplast. So this is in plants. So the DNA found in the chloroplast, we call it chloroplastic DNA. Alright, so that's DNA. And then we have RNA. Okay, so of course, once again, with RNA there are 3 types, and looking over at RNA, the first RNA you have is what we call mRNA, which is messenger RNA. Okay, so that's messenger ribonucleic acid in full, if you want to write it down that way. So it's mRNA. So that's the first one you have, and then you have what is called tRNA, and this is what we call transfer RNA, or transfer ribonucleic acid, if you want to say it that way. And then the last but not least you have is ribosomal RNA. Do not write this in short as rRNA, it will not be accepted as correct. So that one is ribosomal RNA. Do not write it in short. Alright, So I've tortured you with a lot of stuff. I know it's painful, but here we go. A little summary: RNA and DNA. Okay, these are the two nucleic acids, and remember, nucleic acids are built up of what are called nucleotides. So DNA is built up of a DNA nucleotide. RNA is built up of an RNA nucleotide. We remember the differences, right? DNA is said to be double-stranded, RNA is single-stranded, right? DNA contains adenine, guanine, cytosine, and thymine, while RNA then has adenine, guanine, cytosine, but instead of thymine, it now has uracil. This we remember. Pretty simple, pretty sick. So those are the 4 nitrogenous bases, and remember that there's complementary bases, which is the specific ways that we use to pair these with one another. Okay, and we still remember cute girls are all taken. C with G, A with T. Alright. RNA has similar nitrogenous bases. We said this, but instead of thymine, it is uracil. Okay, and the last point we spoke about are the types of DNA. Okay, we have DNA found in the nucleus, we have DNA found in the mitochondria, and we also have DNA found in the chloroplast, and RNA as well. Remember that we have RNA, we have mRNA, tRNA, and ribosomal RNA. That's what we remember. Alright, so that's everything in a condensed package. I'll see you in the next one.